Good afternoon, everyone, and thank you for joining us today. My name is Josie, and I'm part of the public engagement team here at Alzheimer's Research UK. And I'm delighted to welcome you to the 14th event in our Lab Notes online series. Today, we are hearing from researchers from Imperial College London. They will share their research into sleep abnormalities and the links to Alzheimer's disease. But first, I'll go over some housekeeping. So during the event today, you're welcome to switch on the automatic subtitles using the CC button at the bottom of your screen. They're not 100% accurate during the live event, but they will be edited so they are correct on the event recording. If you've missed previous events, you can watch them back in your own time as they are all available on the Lab Notes webpage or on our YouTube channel. During this event, if you would like to ask a question, you need to click the Q&A button at the bottom of your screen, which will bring up the Q&A box where you can type your question. Please do submit questions throughout the event, and we will answer as many as we can in the Q&A sessions after the talks. So now we're going to ask you a few poll questions, because while we can't see you due to the online format of the event, we're really keen to know a little bit more about you. So hopefully those poll questions will be appearing shortly for you to answer for us. So we just wanted to know, how would you rate your knowledge of dementia research? Which of these answers best describes your reasons for attending today? And is this your first dementia research event, or the first event that of our Lab Notes series that you've been to? So I will just give you a few seconds to answer some of those questions for us. And then I'll share those results back with you. So I just give, hopefully you should be able to see, perfect, should be able to see those results on your screen now. So most of you rating that your knowledge of dementia research is average. That's fantastic. Hopefully everyone attending today will learn something new about sleep and dementia, and that a lot of you have a friend or family member that has dementia. So we're really pleased to have you here, and hopefully you can find out a bit more about dementia. And for a few of you, this is most of you, it's your first event, and so really welcome. We hope you enjoy it, and welcome back to those of you that have been before. And just before we start, I've just got a couple of quick slides that I'd like to share with you about what we've been up to at the charity. So I'll just share my screen with you. So hopefully you can see my slides now. And as the countdown to Christmas begins, I'd just like to really quickly tell you about how you can get involved in some fundraising. So you can check out our fundraising page on our website, where you can get a free Christmas fundraising pack to help your fundraising activities over this festive period. So whether you choose to hold a bake-off style competition, or maybe join in on our Christmas pajama day, your efforts and donations will help to make breakthroughs possible. And without further ado, let's introduce our speakers. So at today's event, we are hearing from researchers in the Imperial College Research Network. This network is one of 15 that we support across the UK, providing funding for research, networking and collaboration, and also helping researchers to share their discoveries and progress with the public. Our speakers today will be talking about the links between sleep and dementia. They will share their research into whether sleep abnormalities contribute to Alzheimer's, or whether they may be a consequence of brain changes that have occurred in the disease. We'll be hearing from both Dr. Mathieu Nollet and Dr. Berta and Anuncibay Soto today. Once they've given their talks, I will return to the screen and we'll move to the Q&A session. So you can submit questions at any point during the event, and we'll try to answer as many as we can in the time that we have. I would just like to remind you that Berta and Mathieu are experts in their field of research, but they are not clinicians and cannot answer questions about your health. If you do have concerns about your health or the health of a loved one, please speak to your GP. So without further ado, it's my pleasure to introduce our speakers, Dr. Berta and Anuncibay Soto and Dr. Mathieu Nollet. Over to you.
Good afternoon to everybody. I am Berta and I am a research assistant in Imperial College in Wisdom and France lab. We are a sleep lab, and together with my colleague Mathieu, we are trying to understand what are the links between sleep and dementia.
Hello, everybody. It's a pleasure to talk to you today. So as Berta mentioned, yes, I'm a colleague of Berta, working with her on this important subject. So working at Imperial in the laboratory of Professor William Wisden and Professor Nick Franks in the framework of the Dementia Research Institute. And I will now start sharing my screen. So hopefully you can all see my screen now. So I am really delighted to have this the opportunity to speak today and present and share what Berta and I are doing in our lab to tackle the, I think, important question of sleep disturbances in dementia, and in particular in Alzheimer's disease. So to understand if sleep alterations are an early symptom of Alzheimer's, a risk factor, or both at the same time. So during this presentation, Berta and I will address several aspects of our research within the Dementia Research Institute, or DRI. But first of all, we will briefly introduce what is the DRI and how it operates. So then we will try to explain why it is important to study sleep in the context of dementia, what type of sleep alterations are associated with dementia, and how mice models can help us understand sleep alterations in dementia. And finally, we will present some results of our research projects, which consist basically in trying to answer two important questions. So can poor sleep lead to Alzheimer's disease? And is sleep disturbance an early symptom of Alzheimer's disease? So the DRI recently produced a very interesting short video explaining very clearly what the DRI is, how it is organized, and how it aims to make important discoveries regarding dementia. So instead of me trying to laboriously describe what the DRI is, there it is.
Dementia is one of the toughest health challenges of our time. Affecting more than 45 million people worldwide, a new case is diagnosed every 3 seconds. Thanks to £290 million from founding funders, the UK Dementia Research Institute was launched in 2017 to revolutionise what we know and can do about dementia. We're here to build understanding so that we can diagnose, treat, and ultimately prevent dementia. The UK DRI is not a traditional research institute. We have 7 centres at leading universities across the UK, each bringing unique expertise, supported and connected by a dedicated headquarters team. We're working across the spectrum of diseases that give rise to dementia, all the way from cells to circuits, to systems, to find much-needed treatments. And by probing the earlier signs of disease, we will find a way to stop dementia before it's even started. At the same time, it's important we find the best ways to care for people today by harnessing the latest technologies. To achieve all of this, our researchers share ideas, techniques, and data. There is a vast gap in our knowledge of dementia, and we need to fill it by growing the task force and investing in technology, infrastructure, and ideas. We think differently. Our hundreds of researchers work as one, all to improve the quality of life for everyone touched by dementia. Our doors are open. Come on in.
So as you just heard, the mission of the DRI is to find ways to diagnose, treat, and prevent dementia. And this is an important challenge because around 850,000 people in the UK have dementia, and the number will grow as the population ages. So in 2015, dementia overtook heart disease as the leading cause of death in England and Wales. It represents more than 11% of all recorded deaths. And there is currently no cure for any of the neurodegenerative conditions that give rise to dementia. So I think we are all more or less familiar with the concept of dementia, but what do we really mean by dementia? So there are many types of dementia that encompass different kinds of conditions, because dementia is a syndrome or a group of related symptoms associated with an ongoing decline of brain functioning that can deteriorate memory, thinking speed, mental sharpness and quickness, language, understanding and judgment, mood, movement, and finally sleep. So the link between sleep and dementia has been recently highlighted by a very interesting study of the association between sleep duration and incidence of dementia using data from almost 8,000 participants during 25 years. So they found that persistent short sleep duration, 6 hours or less, between the age of 50 and 70, is associated with up to a 30% increased risk of dementia, compared with normal sleep duration, 7 hours or more, independently of social, demographic, behavioral, cardiometabolic, and mental health factors. So on the graph, on the bottom, you have sleep duration, and on the left, you have the risk of developing dementia. And what we see is that the less we sleep, the more the risk is, reaching 30% at 4 hours of sleep. So I insist here that on the fact that the risk is increased with persistent short sleep duration, which means years of poor sleep and not days or weeks of sleep problems. But before talking about sleep disturbances and dementia, I think it's important to define what is sleep, or at least try to define what is sleep, because actually we still don't know much about sleep. So what we know is that sleep is a fundamental biological process observed widely in the animal kingdom. So most animals and virtually all mammals sleep. And you can see, you can see on the diagram the different lengths of sleep for different species, ranging from 3 hours for a horse and 20 hours for a bat. So if all animals sleep, it must be important. And we also know that sleep is important because acute sleep deprivation can be very detrimental for the organism. However, why and how we sleep, the fundamental function or functions of sleep remain poorly understood. So there are 3 distinct global brain activity states. So the first one is wakefulness, characterized by varying amount of alertness and attentiveness. Hopefully the state you are all in at the moment, if I'm not too boring. Then you have non-rapid eye movement sleep, or non-REM sleep, characterized by unconsciousness and a relaxed muscle tone. So this is what we commonly call deep sleep. And finally, rapid eye movement sleep, characterized by dreams, muscle atonia, and saccadic eye movements. So this is what we usually call light sleep. So the term rapid eye movement comes from the fact that during this phase, our eyes are moving and rolling, as you can see on the video. And in humans, non-REM sleep is divided into 4 substages, and a sleep cycle usually regroup an episode of non-REM sleep and an episode of REM sleep. And we usually sleep during the night in one block. This is why it is called monophasic sleep. So in rodents, sleep is more fragmented and distributed throughout the day with many small sleep cycles. So this is why we say sleep in rodents is polyphasic. So how do we measure sleep? So there are 3 main biomarkers of sleep. So the first one is the electroencephalogram, or EEG, which measures the brain electrical activity. The second one is the electromyogram, or EMG, that monitors muscle tone. And finally, you have the electrooculogram, or EOG, which measures the eye movements in humans. So the EEG is usually measured using electrodes on the scalp, both in humans and in animals. So the 3 brain activity states, so wakefulness, non-REM sleep, and REM sleep, are characterized by different electrical activity of the neurons, as you can see on the picture. And by looking at the frequency and the amplitude of the EEG, we can then classify sleep into wakefulness, non-REM sleep, and REM sleep. This is how we measure sleep. So after that, we can create what we call a hypnogram. So in humans and in rodents, using the classified EEG traces. So the hypnogram being basically a graph representing the sleep stages as a function of time. So on the right hand side, you can see that this person is awake and then goes to non-REM sleep 1, 2, 3, and 4, then REM sleep, then again and again non-REM sleep followed by REM sleep until the person wakes up 7 hours later. So now I think you know as much as us about sleep, and it is time to talk about sleep in dementia, in Alzheimer's disease in particular, and Berta will take over from here.
Hi, everybody. I am going to try to link what is the aspect in which the sleep start to link with the dementia or the Alzheimer's disease in particular. We noticed like a decade ago that around 70% of the patients that will develop cognitive symptoms in the future, before they develop any cognitive impairment, they used to develop some sleep alterations or sleep disorders. These sleep disorders are really diverse, and here in the slide I highlight one of the most important or most common that we see. The first one is what we call sleep fragmentation, that this is really well known as insomnia. This is characterized usually by, instead of having this nice episode of sleep that Mathieu just explained to you, that you have a block long of non-REM and then you go to REM, this long episode that they are long in the people that they don't present any disorder start to be interrupted by wakefulness. So the brain activity change because they go to wake when they are not supposed to go to wake. I would like to highlight that sleep is a very complex process. It can appear that the sleep is a passive process, that in which we lie in a bed and we don't do nothing more. And it's true, but in our brain, the activity is changing a lot, and it's a really active process that happens because a lot of our brain areas are connected by wires, that these are the neurons, that they connect different brain areas. So the sleep is something that can be easily affected when our cells in the brain, our neurons, are starting to accumulate substance inside. And this is the reason, because we start to see all these sleep disorders, like the insomnia. The people is not able to keep what we call a consolidated sleep, a good sleep, long enough. Also, it's very common that they start to nap during day. This is because the brain is getting confused. These wires are starting to be confused, and instead of sleeping when it's normal in humans, as Mathieu explained to you, in darkness, because we need to segregate certain hormones to fall asleep, like melatonin, we start to sleep during day because our wires are starting to be confused, are starting to accumulate certain proteins that I will go into this later. So this make our brain to work not properly. And this is the reason, because the sleep start to change. We saw a decrease in the time of sleep, the time that we are in non-REM or in REM. And as Mathieu explained to you, we also saw a decrease in the potency of the sleep. These frequencies, this activity of the brain, this electrical activity start to change. The amplitudes and the frequencies change. This usually means that the sleep is less efficient. And now in the last 10 years, we are realizing that the sleep is really important for a lot of processes. One of them is the dementia, because the sleep is proposed to be a clearance system from the brain. So it is supposed that when we sleep, we are clearing substance. So if we reduce the quantity of the sleep and the potency, in a way, we are accumulating a lot of waste in our brain. But this can be simplified to two simple questions that we don't have the answer. And these are two questions that Mathieu and me we are trying to solve. What is the important thing? The quantity, the number of hours that we sleep, independently of the quality, how these episodes of sleep they are really long, how these waves they are, or is the quality? Doesn't matter if we sleep short episodes of sleep, but if they are correct, if they are good enough, or what is important for the dementia pathology. But still, we don't have the answer to these two questions. Next slide, please. So what is happening? Sorry, what is happening inside of our brain in the Alzheimer's disease is that two proteins, they are starting to accumulate in our brain. As Mathieu explained to you, Alzheimer's is a kind of dementia, but this is the most common dementia. And at the same time, it's a more aggressive dementia than the normal dementia that is related with aging. It happened before and it happened faster. And this is the reason, because we start to see all these sleep disorders, because inside of our brain is happening something. Two proteins, these are the most important proteins in the Alzheimer's disease, that they should be there, they are useful, and our cells need these proteins for work properly. They start to aggregate. And probably you hear before about amyloid plaques and about tau tangles. Tau is a protein that is part of the skeleton of the cell, so needs to be there. And amyloid is a protein that we need to make all this connectivity of our brain. So these proteins, they are segregated every day in our brains, and they are clear every day while we sleep. But there is a moment that the patients of dementia start to accumulate these proteins. Next slide, please. And this is what we show here. And this is how finally we can say that a patient have dementia, because before the medical doctor, they diagnosed them due to a battery of tests. But the way to really prove that this is Alzheimer is to take a piece of the brain of this person and to perform an histology, that this is what you are seeing here in the purple image, and stain the cells for certain colorants that will show us if there are any deposits in these cells of amyloid, that are the pink round deposit that you see, or of tau, that are the skeleton of the cell, that are these flame shapes that you see. This is not present in a normal brain. This is present in a brain that suffers from Alzheimer's disease. And during years, these proteins are accumulating inside or outside the cells. This is the reason, because the sleep is affected, because the neurons cannot work properly, because they are accumulating things inside. Another thing that we can see really easy for a patient from dementia is that the brain shrinks. The brain, instead of having the normal shape, become tinier, probably due to this accumulation. But this is the information that we had until recent years, because we were able only to take this information from the endpoint when the patient sadly died. So we don't know what is happening before. And this is the reason, because we are not able to cure or to treat dementia right now, because we don't understand we only know the end of the movie, but we don't understand what is happening at the beginning and in the middle of the movie. And this is the reason, because we work with animal models. Next slide, please. They are going to be the one that they are going to help us to cure this pathology. Because even if they look really different to us, and they look really different to us, they are really similar to us. The truth is that they share with us a 95% of their DNA. So they can develop a pathology, and Alzheimer's disease in particular, really well and really similar to a human. So they will help us to understand what is happening before and what is happening in the middle. So we will be able to understand what we need to stop or what we need to increase to stop this pathology or to delay this pathology. This is the reason, because until now we were unable to do this, because we didn't have chronic models of sleep disorders, and proper models of dementia that allow us to understand the whole story. Next slide, please. And here I would like to show to you really quickly how similar they are and why we can compare them with us. As Mathieu explained to you, in the sleep, they are really, really similar to us. This slide is trying to highlight the difference that they have from us. They sleep mainly in the light phase instead of in the dark phase. They are polyphasic, as Mathieu told you. I think this is really easy to understand. If you have a dog or a cat at home, you will realize that they sleep a lot during the day. They take really short naps, but a lot of naps. So at the end, this results that their episodes are shorter than our episodes, but they sleep more during the day. But the rest is really equal to us. The potency, the amplitudes, the frequencies, the episodes. So they are a perfect model to really allow us to understand what is the importance of sleep. Next slide, please. Also, they are really good models now that we have the proper ones for the pathology, because as I showed to you before in a human tissue, this is what we are able to see in a mouse tissue. And you will see again this pink deposit, that is this amyloid aggregating in the brain of the animals, or tau, that is the image in the middle. Here you see 3 columns. The one that is more in the left is a normal mouse. The one that is in the middle is a mouse that is accumulating tau. And the one that is more in the right is a human. So you will see that the mouse and the human present a very similar staining. This means that they are precipitating this protein in a very similar way. And also in the right hand, you can see an MRI scan of 2 animals. The upper part is a normal animal, an animal that doesn't present Alzheimer's disease. And the down animal is an animal that presents Alzheimer's disease. And I highlight some of the brain structures with dotted lines. So you can see that these structures are tinier because they are also shrinking, like in the humans. Next slide, please. And press play, please. This is a very short video to show you that, of course, this is the consequence that all of this have in the animals is the same consequence that we have. We are going to see 2 animals swimming in a pool. They are trained to swim in this pool and to find a platform. This first animal is accumulating tau. So even if he's trained to find this white platform that you will see in the right of the screen, he's not able to find it because he doesn't remember where is the platform. So he needs to swim around the base to try to find the platform. And even if he goes really quick, really close to the platform, sometimes he's not able to find it. And now you are going to see a second mouse that is not accumulating tau and is also trained to find the platform. Can you see how quickly he do it? So they recapitulate really well all these features that we have. And of course, they will allow us to understand all the intermediate steps that we cannot understand in a patient. Next slide, please. So as Mathieu told you, next slide, in our lab, we are trying to answer 2 very simple questions that is incredible, but still they don't have an answer. What is all these sleep disorders? So when we are awake, as I explained you before, we are segregating all these proteins that are needed for the brain connectivity. So in a way, we are accumulating these proteins. And when we sleep, we clear these proteins. So if we stay more time awake, we are creating, next slide, please, a perfect scenario in our brain to develop dementia. So maybe the sleep is a risk factor and can explain some part of the dementias or the Alzheimer's disease. Or can be only a very early symptom. But if it's a very early symptom, it's a very interesting symptom because it happened before we have any cognitive impairment. This means that probably is saying to us, this is the correct window to apply a treatment because we don't have still cognitive symptoms. Next slide, please. So as I mentioned to you before, these are results from our lab. We couldn't understand this before because sleep is so fundamental, as Mathieu explained to you, that if we deprive an animal chronically to sleep in a traditional way, or we don't allow the animal to sleep, the end is going to not be beautiful because it's something that we need to do, like eat. So we really need to understand really well the connectivity of the brain to only manipulate certain populations of the cells to have some animals that present some sleep disorders, but are mild sleep disorders. And at the same time, in the rest of the things, they are completely normal. So they mimic perfectly the persons that they are not able to sleep properly, but in the rest, they are completely normal. And we create 2 different models to try to answer the 2 questions that I put at the beginning. What is the important thing? The quality of or the quantity? So we create a model of animals that develop insomnia, touching a very few cells in the brain of the animal, that is the ones that you see more bright in the image down. I would like to highlight that this is not all the cells that there are in this brain area. This is only a subtype of cells that are really important for the sleep. This is the reason because we can work with them during a lot of years because they are perfectly normal animals. They only have certain alterations in the sleep. And the other image show another group of cells that is also in green, that is a very tiny group of cells that are also really important to explain the time that we are awake. when you modify these neurons, you result in around a 30% more time awake. So these two models will allow us to understand what is important here and what we need to take care. Next slide, please. And what we did with this animal, that is the first one that you have in the left hand of the screen, is put together with an animal that is the one that is called APP and LGF, that Mathieu will explain you later more, that develop dementia in a very slow way, very similar to the humans, because as you know, the dementia is a progressive disease. Alzheimer's disease is a progressive disease, needs a lot of time, goes really, really slow. So we put all these two animals together, and now we have two types of animals. One that present insomnia, but will naturally accumulate amyloid as the humans, and other animals that have a little increase in the wakefulness. So they are around a 30% more time awake than a normal animal, and they will also accumulate amyloid. So we will be able to identify what is important. Next slide, please. Here, I would like to show you really quick how we see this. So in these graphs, you are seeing the measurements of sleep of control animals, that are the black bars, with animals that they are sleeping a 30% less, these ones that they present this sleep loss. And we measure them in different moments during our research, because we keep them really long and we work with them really, really long. And as you will see in the graphs, the purple bars, they present a reduction in the time that they are sleeping, especially in the dark period. This is the reason, because this part of the graph is a little darker, because it's the moment that they are sleeping less. But if you see the light period, the part of the graph that is completely white, in this moment, they are sleeping in the same amount that the control animals. So it's not that these animals, they are not sleeping, they are sleeping a little less. They are like a person that sleeps 5 hours every day. And of course, as you will see in the other graph, this results in an increase of the total time that they are awake. And this is chronic. This stays in them as they age. Next slide, please. And now we are starting to analyze, after years of this, to analyze the amyloid burden that these animals present. Here you can see two brain tissues that they come. The one that is under the purple title is the one that was chronically presenting a sleep loss. And the other is a normal animal, it's a control animal, was the colleague. So what we saw is that the animals that they were losing chronically sleep, they present more amyloid plaques in different brain areas, that this is what is shown in the graph, than the controls. And now I am going to leave you with Mathieu, that will go more in detail about this APP model.
Thank you very much, Berta. So as Berta just showed, poor sleep can lead to amyloid beta pathology. But as she also mentioned, amyloid beta naturally exists in the brain independently of any disease. It's just that you have more amyloid beta in Alzheimer's disease that generate plaques after a certain period of time. So what has been observed is that the release of amyloid beta is different during wakefulness and during sleep. So in humans, the level of amyloid beta is increased during wakefulness and decreased during sleep. We observe the same thing in rodent, so an increase of amyloid beta when the animal is awake and a decrease when the animal is sleeping. And when an animal is sleep deprived, so kept awake longer, there is an increase of amyloid beta. So sleep affects the release of amyloid beta. But it has also been observed that an animal model of Alzheimer's disease, so which has naturally a lot more of amyloid beta, displays sleep alterations such as an increase of wakefulness, a decrease of REM sleep, and a decrease of non-REM sleep. So it seems that amyloid beta influences sleep architecture. Clearly, there is a strong link between amyloid beta and sleep. But the question is, does the slow accumulation of amyloid beta before any cognitive symptom can cause sleep alteration? And in other words, can sleep changes be seen as an early symptom of Alzheimer's disease? And to answer this question, we have two strategies. So the first strategy is the study of sleep and sleep-related alterations in a special transgenic mice, codename APP NLGF, doesn't really matter, displaying slow accumulation of amyloid beta pathology in all the brain. So for this, we simply record sleep at several time points for 1 year in these animals, and I will show you some results now. So first, if we look at what is happening in terms of total sleep time during the light period, which is the rest period for the mouse, we don't see any difference between the control normal animals in gray and the animal with higher level of amyloid beta in red. So both for female mice on the top and male mice on the bottom at 8 months of age. But at 12 months of age, female and male mice with amyloid beta sleep less than normal control mice. So the star here means that there is a significant difference between the two groups. In female mice and in male mice. However, during the dark period, so the active period for the mouse, we don't observe a change of total sleep time. And interestingly, when we look at REM sleep, so already at 8 months of age, we observe a significant decrease of REM sleep in female and male mice with higher level of amyloid beta during the light period. So the decrease is even stronger at 12 months of age. And in the dark period, the active phase of the animal, we only observe a decrease of REM sleep at 8 months of age, but we continue to our analysis, and perhaps with bigger experimental group, we will see differences at 12 months of age. So, okay, it seems that slow accumulation of amyloid beta influences sleep. But here, amyloid beta is present in all the brain, and perhaps there are specific brain regions that are particularly vulnerable to amyloid beta and sleep regulation. So this is why I am particularly interested by the cholinergic system in the brain responsible for the release of acetylcholine. So acetylcholine is a neurotransmitter secreted by cholinergic neurons in the brain. So these cholinergic neurons of the basal forebrain, so in green on the picture here, are particularly vulnerable to neuropathology in Alzheimer's disease. And interestingly, a type of drugs called cholinesterase inhibitor can increase acetylcholine levels and partially ameliorate cognitive symptoms and enhance quality of life for patients with mild to severe Alzheimer's disease. And finally, cholinergic neurons of the medial septum, so just here, orchestrate the regulation of REM sleep. So it seems that the cholinergic neuronal population of the medial septum is an interesting target to investigate in the context of Alzheimer's disease and sleep. So the second strategy that we are using to investigate how amyloid beta affects sleep, especially in relation with the medial septum, is the study of sleep and sleep-related alterations in special transgenic mice, codename MS-chat APP and LGF, again, doesn't really matter. And these mice display amyloid beta pathology only in cholinergic neurons of the medial septum. So for that, we target the medial septum with a chemically engineered substance that will make the cholinergic neurons of this area produce more amyloid beta only in these neurons and only in these brain regions. First of all, I only show the results for male mice here. We are currently collecting the data for female animals. So, and what we observe is that in terms of total sleep time, there is no difference between the control normal animals in gray and the animal with amyloid beta in the medial septum in red. So 8 or 12 months after surgery and during the light and dark period. However, the time spent in REM sleep is significantly shorter in animals with amyloid beta, specifically in the medial septum, compared to control animals in gray. So it happens in the light and in the dark, and 8 months and 12 months after the surgery. So here we observe a very strong effect of the presence of amyloid beta in the medial septum on REM sleep. Sorry. So to conclude, our data seem to indicate that chronic sleep loss can increase amyloid beta pathology in the brain. Also, slow accumulation of amyloid beta in the brain can alter sleep and especially induces a decrease of REM sleep. Furthermore, cholinergic neurons in the medial septum seem vulnerable to amyloid beta pathology, and again, this leads to a decrease of REM sleep. So perhaps, hopefully in the near future, by improving sleep disturbances, we might be able to delay or prevent more severe symptoms of Alzheimer's disease. So this is really a teamwork. And before finishing this presentation, we would like to thank our supervisors and lab mates for the precious help every day. So this is a screenshot that we taken a bit more than 1 year ago during lockdown, when we were trying to all work hard with difficult access to the lab. Also, I have to say that nothing would be possible without the support of the DRI and all the funding bodies. And finally, thank you all for your attention and your interest for our work today. And I think now it is time for the Q&A session led by Josie. Thank you very much.
Thank you, Mathieu and Berta, for those really fascinating talks. They were great. Thank you so much. So we've had quite a few questions come in the Q&A so far. So I'll start off sort of combining a couple maybe. So if we could improve sleep for patients with or humans with Alzheimer's, could it be reversed? So I think you've talked about it in mice, but could this be applied to humans or not? Maybe Berta, we could start with you for that one.
I think this is the point that we are starting to get more clear right now. We know for sure that this cannot harm, and there is a lot of reasons because can be good. So it's true that it's tricky because usually the drugs that help you to sleep, they are not thought to be taken, not all of them, chronically, but a lot of therapy is going in this direction, and there are even some clinical studies that they are using some drug for sleep, try to especially slow down the process.
Mm-hmm.
Yeah, I have nothing to add. I think Berta said everything. So yeah, indeed, it's difficult to answer because it's actually what we are trying to do and what we are trying to understand. And she's right. There's actually like 1 or 2 clinical trials trying to use pills that makes you sleep better to try to and see if it prevents, but it's very long, because the problem with Alzheimer's disease is that it's a very, very long process, and the time to accumulate data and the research is also very long. So but we hope to achieve that one day. Yes.
Thank you. Another question from Richard, who asks, have any trials been done to see if induced sleep clears amyloids or tangles, or do you know anything about that? Mathieu, maybe one for you.
That's a very good question. Actually, this is also something we are trying to do in the lab, because there's actually a lot of debate in the scientific field if really sleep has an effect on the clearance of all the byproduct of the neurons and of the amyloid beta and tau. Berta explained very well. So this is not very clear. I mean, yeah, you have some theories mentioning that actually sleep can have an effect on this aspect, but some groups, and I will try not say too many things, because this is ongoing research. I mean, the results are not that clear. So yeah, we are trying to understand this aspect. So but this is a very interesting point. Yeah.
Yeah, because it's true that it's not easy, because if you sleep less, the result is that you are more time awake. So you are accumulating more of these proteins naturally. So maybe it's true that the sleep can clear molecules, or maybe the key part is that you are not awake enough to accumulate them.
Mm-hmm.
So, but this is the part that we don't have clear, but we know for sure that more time awake means more accumulation. We don't know how, but this is the reason, because before I say to you, yes, I think sleep will help in whatever the cause. We don't know why, but will help.
Mm-hmm. Great, thank you both. We've had another question that says, how does your studies tie in with studies interlinks with menopause and dementia, as sleep disturbances can be a symptom of menopause? I was wondering if either of you knew anything about that.
I mean, I get we are not. I mean, this is not the question that we are focusing on, but what we are doing is looking at what is happening in male and female. I'm not a specialist of menopause, and this is not my field of expertise. But I mean, this is something we take into account. I mean, the difference of sleep changes and accumulation of amyloid beta if you are in male and female, because what was lacking from previous studies is the use of female mice. Actually, a lot of things are done in male animals in general. But we do understand, and we do know now that you have some differences in terms of the vulnerability of Alzheimer between men and women. So this is something that we are trying to understand. Yeah.
Thank you. We've had a question from Mary. Would it be fair to theorize that it is the REM phase of sleep that is the most responsible for clearing amyloid and tau from the brain?
Oof, this is a very I mean, it's difficult because REM is really short, and the truth is that a lot of this research is mainly they talk about non-REM. When they talk about the sleep, they are talking about non-REM because it's the 80% of your sleep. And we are trying, and this is also something that we are trying to do in our lab during years, try to identify when you enter in a REM sleep, and then or promote more sleep or promote wakefulness to break this episode. But it's really difficult technically, and I think right now we don't have the technology to answer properly this question.
Yeah, exactly. I mean, there's differences between non-REM sleep and REM sleep. Even if we don't know everything about sleep, we know few things. And I mean, when we sleep, non-REM sleep will be more involved in the healing of the body, like the energy, while REM sleep is more linked to the memory or the regulation of the mood. So saying that REM sleep actually has a more beneficial effect in the clearance, that's something, as Berta said, we don't know, and it's difficult to do because especially in animals, when they have very small sleep cycles, the REM sleep episode can be very, very short. And it's difficult to imagine that during the few seconds of REM sleep, a lot of things might happening in terms of brain clearance. But who knows? I mean, we are trying to tackle this problem in our lab indeed, but it's technically it's difficult.
Thank you. We've had a couple of similar questions about sleep patterns for shift workers. So is the incidence of dementia higher in shift workers who have abnormal sleep patterns? And another one, shift workers have poor sleep for hours and years. Can this cause accelerated damage, and can it be reversed? So would either of you like to answer something about that, shift patterns?
Yeah, no, the answer of this is yes. And even the paper that Mathieu showed that make the magical number of 7 hours, one of the very important groups that they were in this article was shift workers, because they need to change their sleep. So this is true. When you change the patterns of the sleep, there is an increased risk of develop dementia. So I am sorry about the people that they do shift works, but the answer is yes.
There's clearly links between shift workers and poor sleep and dementia. The thing is that we are talking about a risk factor. So it's not because you have like a problem of sleep that you will necessarily trigger and necessarily have a problem with dementia. This is a risk factor, so it depends on so many factors, so many things. We are also very different sleepers. I mean, some people, they need more sleep. Some people, they are perfectly fine with less sleep. I think that the key aspect is that when you wake up, do you feel rested? Because you can feel perfectly fine with 5 hours of sleep and then have any problem without you don't have any problem, and then you can also sleep more, but you don't feel rested, and then you also have problem. And the thing is that it's always there's a difference between the subjective sleep and objective sleep. Sometimes people, they feel that they don't sleep well while they actually sleep okay, and the other way around, people, they don't think that they have any problem while actually they are waking up regularly and the sleep is fragmented, but they don't really realize. So it's something, it's a difficult subject, but in general, I mean, yes, I would say if you have a better sleep, then you have less risk to develop any type of dementia.
That probably answers the question that I was going to ask next, which is about sort of sleep disturbances, and does waking up to use the bathroom but then going back to sleep and having 7 hours in total and feel like you're sleeping well, does that count as a sleep disturbance? Like what
No.
would you consider disrupted? Yeah, no.
No, even if it's what Mathieu say, this is a really true thing. If you wake up feeling rest, means that for your brain is enough, and we are different. This is only risk factor. Of course, it's like the tobacco. The tobacco increase the risk factor to develop a heart infarct. You will have a heart infarct for sure. No, but you are increasing the possibilities.
Yeah, thank you. We've had a question from Tracy. So for patients with narcolepsy, do you know if there's an increased risk of Alzheimer's in this group?
Maybe Mathieu can help. This I cannot say anything.
Actually, this is a good question. I think yes. I'm not 100% sure because I studied narcolepsy, but years ago, and not in linked with actually with Alzheimer, more linked with depression actually. But and again the link with sleep and depression and stress is also something very, very interesting. But patient with narcolepsy, I don't want to say something silly or to say something wrong. Might be something because it's the narcolepsy is usually most of the time induced by a disturbance of something called orexin, the orexinergic system in the brain, which has actually a lot of different effects on the brain. And there's also a link between this orexinergic system and deterioration of this orexinergic system and dementia. So I don't know if there's direct data between narcolepsy and dementia, but I think that there's it's possible.
Thank you, Mathieu. So a question from Denise, who's asking about mice being polyphasic in their sleep patterns, whereas humans are monophasic, and sleep disturbances in humans would turn their pattern more polyphasic. So could we use models that are monophasic, such like we are as humans? And she says, in a way, the normal sleep pattern in mouse is akin to the pattern in dementia patients. So maybe to answer that question, and maybe then ask a bit more, my question would be like, what are the positives and negatives of using a mouse model for this kind of research as well?
Of course, I mean, you have differences between sleep in humans and sleep in rodents. You have things that you can perfectly do in when you study sleep in humans, but some of the things you can't. So you need to find a model, and there is not a lot of options, to be honest. I think that rodents in general, even if they are not monophasic, it's not that important, what I want to say, because we know the difference, we know how to analyze these differences, and then when we analyze our data, we take into account this difference, because basically, apart from few differences in the length of sleeping and the number of sleep cycles during the resting period, the rest is pretty similar. So I think that there are a lot of investigation in humans in terms of sleep and Alzheimer. It's just that you need a different setting. You can discover things with human that you can't do with a mouse, and the other way around. I mean, we need to keep in mind that the ultimate goal is not to study Alzheimer in a mouse. I mean, this is just a model. What we want is to be able to translate these discoveries to humans. And that's why we are doing things in discovering things in a mouse model. And then we can to apply what we discover in humans, trying to do some other things, trying different type of drugs and or different therapies, and this is how we actually make progress. So we need to start somewhere, and I think mouse is actually a very good model.
Yeah, and I will say that these differences, as Mathieu say, are parameters that we can control. So we know how to control them, we know how to minimize all this impact, and is the only source that we can get information. The other option is accept that as the population age, we will increase the percentage of Alzheimer or dementia. So this is the reason because we do this. It's not for study dementia in a mouse, it's for really translate it to the life of real people.
Thank you both. I think we've probably got time for just 2 more questions. So the penultimate one, I'm curious about how you classify sleep stages, Joni asks. Is it done automatically? One expert, multiple experts? And another question, have you focused on other sleep stages besides REM, N1, N2 or N3?
This is a great question because we spend, and Mathieu will go later, I am sure in detail about this, we spend a lot of time of our life analyzing this. This is not manual. I mean, it's true that we have algorithms that they put some states in the sleep, but I will say to you that analyze one day of sleep of an animal take a couple of 3 hours. So it's the most of the time that we spend is scoring these files and trying to understand how these animals sleep that night.
Yeah, and what Berta forgot to mention is that we have a lot of data to analyze. So it's a very, very good question. You indeed, I mean, you have some algorithm to automatically classify sleep and classify the sleep stages. The only problem is that what we collect, the data we collect from the EEG is a biological signal. So you might have some artifacts, you might have some problem when you collect the data because you are collecting data in a biological organism, and then you have some lot of differences between the EEG you collect from one animal to another. And the problem is that sometimes algorithms, they have some problem to adapt to the specific aspect of sleep from one animal to another. So they are good tools. We have some good tools, but the reliability of these tools sometimes is a bit low. This is why the gold standard at the moment is to have 1 expert, 2 experts looking at the EEG trace and yeah, just classify and analyze the sleep this way. So I mean, yes, we work usually in the lab, we work blind, so we don't know which when we score, when we do it, we don't know which animal it is, and we usually have also some other people that they can also have a look at our scoring, just to be able to make sure that actually we agree between several experts of sleep. And for the question about REM sleep and non-REM sleep, what we investigate, of course, we are looking at actually everything. What we presented today is just like a very small part of what we observe. I focused personally on REM sleep because it's where I have the most interesting data. But you have also some little changes in non-REM sleep. But I think it was more interesting to show that actually in previous work, as Berta said, a lot of people focused on non-REM sleep and basically pay little attention to REM sleep. Actually, if you pay attention to REM sleep, you see that there's also a lot of things happening there. So this is the reason why I wanted to focus on REM sleep today. But yes, we are working on all sleep stages.
Thank you. I think we've probably run out of time just for our last question now, so I think we'll have to end it there. And thank you to everyone that submitted a question today. I'd like to round off the event by thanking Mathieu and Berta so much for giving such interesting talks and answering our questions. It's been a pleasure to have you both. So thank you and goodbye to you both. Bye.
Thank you very much. Thank you. That was a pleasure. Thank you.
Yeah, the same. Thank you for inviting us. It's really important for us that the people understand what we are doing every day, because this is the way that we will move forward in these pathologies. Thank you.
Thank you. Thank you everyone for joining. Just before you go, we've got a couple of poll questions that we'd love you to answer, just to give us some insight into how you found today. So hopefully those should be appearing on your slides, on your screen shortly. So the first question, how did you find the content of the talk? So was it too technical, about right, or too simple? Would you recommend our lab notes events to your family and friends? And have you learned something new about dementia research? So I'll just give you a couple of seconds to answer those. And then we'll share those results with you as before. So it's great to see that lots of you, well, most of you thought that the content was about right. That's fabulous. And that you would recommend these events to your friends and family, and that most of you have learned something new about dementia research. That's great. That's just what we want to see. And we'll also be sending a more detailed feedback survey to you soon, and with the video recording likely to come next week. Please do complete the survey, as we'd appreciate any feedback you can give us about these events. We're also gathering input into the 2022 series in the survey, so please do tell us what you'd like to see from next year's Lab Notes events. For example, if there's any particular topics or areas of research you'd like to hear about. And as we finish for today, we have a few more slides to share with you. And that's just to say that our final event of the year will be taking place on Thursday, the 16th of December at 7 p.m. So join our Chief Medical Officer, Professor John Schott, and leading dementia researchers to hear about the latest progress being made for an informal chat and conversation at Christmas. So you can sign up online to join us for this event using the QR code or the link on in the chat. And I also know there were some questions that we weren't able to answer today, and some where you may need more personalised information or guidance on where to turn. So our dementia research info line is there to help, and they can answer questions and signpost to other sources of information and support. So please do get in touch with them if there was any questions you had about today that we weren't able to answer. We also will be having a Q&A next week on Instagram. So if you have any more questions about sleep and dementia specifically, you can join us next week for a live Q&A on our stories. So make sure you're following our Instagram page to join in with that. So you'll be able to submit questions anonymously, and these will then be answered by our expert team here at Alzheimer's Research UK. So do make sure you're following the page to find out more. And I just want to say a huge thank you for joining us today, and for joining us throughout the year as well, if you've joined other events. I hope you found it really interesting and useful, and will join us at our Christmas event or join us for the series next year. Thank you, and goodbye everyone.